All right, guys. I am going to go over the most important things from your lab so that you are solidifying what you should have learned from it. So, Station 1, you should have read this information, and the idea behind Station 1 was comparing two unicellular organisms, except for that one of the unicellular organisms, or a lot of them, were prokaryotic, and then there was only one or two eukaryotes at the other microscope. So we're looking really here at the size between these two, difference between these two unicellular organisms. So here you can kind of see what was on your example screen, and then this is an actual picture from the microscope. Both sides of the classroom looked pretty much the same, so you can see that it's pretty similar to the image here. The one thing that I really wanted you to understand is that, like, this little string right here is a whole lot of individual bacteria in a string. These are all these little dots are individual bacteria. They are way too small to be able to see inside of, so you can't even see what the nucleoid would be or where their DNA is. This is hundreds and hundreds of different bacteria. And the thing about it is that when we're looking at these different cells here, you can tell that here in this field of view, which is 400 times magnified, you have hundreds of bacteria. But in this same size field of view, which is both 400 times magnified, you only have one or two paramecium. These are our eukaryotes. So if I put them side by side, you can see one paramecium is the size of, 100 bacteria. So these are clearly eukaryotes. If you were on the side of the classroom with the classroom doors on my left, then you were looking at this image. If you were on the opposite side of the classroom, then you were looking at this image right here. So again, both of them pretty close to what the example image was. I just really wanted you to be able to see that there's a clear nucleus right here in the center of these little protists. Okay, that's the kingdom that the paramecia belong to. So that's what you were looking at. And then on your answer sheet, this was a picture of a student who I thought did a really good job. Notice that they had the magnification recorded there. They also recorded that the bacteria were prokaryotic. I like how he made little tiny dots and squiggles and did not obviously have a nucleus in that picture, but here the nucleus was clearly drawn and labeled, and he appropriately labeled the bacteria prokaryotes and the paramecium as a eukaryote. Of course, the eukaryote is larger, and the reason that you can't see the inside of the prokaryotes is because they are too small. So let's look at the next station. This next station had two eukaryotic examples, but one of them was a multicellular eukaryote and the other one was a unicellular eukaryote. This was a different unicellular eukaryote than what we saw in the first station. It was an amoeba. If you've ever heard of the monster like the Blob, it is modeled after an amoeba. So amoebas can actually shift their cytoplasm in their body, so they shape change a lot. They kind of drag themselves along whatever surface or whatever water they are in, and engulf their food. They're very large for unicellular organisms, and they have some pretty clear organelles that are easy to see, but still the nucleus is the easiest and largest organelle, easiest to identify. If you were on the door side of the classroom, this is the door side of the classroom amoeba, and it was kind of a compacted, all of its pseudopods, its little feet were all kind of under each other, but you could still see the nucleus, which is why I chose that specimen. And then this one looked a little bit more like the image in your example sheet. So if you were on the opposite side of the classroom, this was the one that you saw. You should have been able to label those nuclei, so the nucleus in this amoeba and the nucleus in this amoeba. This one was a little bit harder because it had just eaten something, so there was something underneath it that was kind of masking the nucleus or looking like a double nucleus, but it was still just one organism there, with just kind of some of these other different organelles and some food that was visible. Both of the animal tissue examples looked very, very similar. This was one of them, and it looks very similar to the example. This one is, I had to shrink it to fit on the thing, but if I make it bigger, then you can see it looks almost exactly like the picture here. And you can see that there's these little bitty purple nuclei in each one of these cells that are all kind of squished up together. They're all kind of a rounded shape, but not a perfect circle like the drawings are, right? But you can clearly see a lot of the nuclei. You can't always see nuclei in tissues that are three-dimensional, like even the amoebas. They're not, a flat layer on a sheet. So some things are in focus and some things aren't. So sometimes you can't see the nucleus in every cell. That doesn't mean they're not there. Okay, they're just not in focus. So that's what you're looking at in that second station. And for the second station, this was, I thought, a really good drawing. They had all of the cells kind of squished up together in the animal cells, and the nucleus clearly labeled in both the animal cell and in the unicellular amoeba. The amoeba, it says, is the amoeba the cell the only part of the amoeba? Yes, it's just one cell. So if it's unicellular, if you're looking at one cell, you're looking at the whole thing. And that's the opposite answer for number 6. Of course, that's just a really small piece of liver tissue, right? So that's just a really small piece of one organism, even though it's a whole bunch of cells, because animals are made, depending on how big they are. Mammals, which is what this came from, either of hundreds or trillions, hundreds of thousands or trillions of cells. So, and how do we know they're both eukaryotes? Because they both have a nucleus. So that's what we're looking at for that station. I'm going to pause this and pick up the last 2 stations in a separate video.